Hello, in this video we will discuss about the epigenetics and mutation. We will discuss about the epigenetic related to the mutation and what is the mutation about epigenetics, and we will discuss. So first of all, we should understand about the definitions. The epigenetic is the study of epigenome, which is above of the genes cause mutation without alter the DNA in the cell. So it means that without changing of the DNA pattern, the insertion of the molecule methyl or methylation is known as the epigenetics. So the advantages and disadvantages of the epigenetic is helpful. Epigenetic is helpful for animal and plant survival. Epigenetics modification is on the basis of changes in environment and development of embryo to whole organism. Some epigenetics changes cause severe mutation to cancer, cancer causing cell due to the rapid growth and uncontrolled cell growth due to the activation of excess of activation of oncogenes, while the suppression of the cancer gene suppression of the tumor suppressing genes, which that will be inactivate. So that was the overview. We will discuss in this video. The whole video is made up of just an overview, so that you can understand the overview, what is the gene, what is the epigenetic. So that is important thing. So that basically our tissue, our whole body contain a same DNA pattern and same genome in every cell, but their changes are due to the epigenetics. For example, the skin cell produce the basically skin cell, the gene is active according to the skin cell, while the liver cell according to the liver hepatocyte and etc. and basically just like that. So why this happening? Why liver cell produce the liver cell? Why muscle cell produce the muscle cell? That is due to the epigenetics. So here is the tissue of muscle, basically is contain a nucleus. So these nucleus contain a chromosome. This chromosome is tightly bound with the nucleosome, and nucleosome is made up of histone protein and tail of the histone, and that is DNA is wrapping around the histone protein. So that is basically histone proteins contain 8 subunit, but their type of histone protein is 4. So you can imagine that. So here is the DNA, which that is, for example, the gene containing DNA, which that is methylation. So this methylation, for example, promoter gene, which that is promote to regulate the gene sequence and transcribe the gene sequence, so the DNMT1, and maintain the division. So the DNA methyltransferase enzyme used to methyl transfer into the DNA segment. So the DNA methylation and modification is occur. So the histone modification is basically here, which that is basically maintain division of cell, embryonic cell, and that is basically embryonic cell is the DNA methyltransferase 3 alpha and DNA methyltransferase 3 beta, which that is during the embryonic cell, while the distinct cell, which that is the distinct cell type, is the enzyme used is known as the DNMT1. So that's different enzyme used according to the different stages of life. So that's important thing is that let's begin to understand how this methylation will affect. So the transcription and translation, in this time we will discuss that. Here is a transcriptional gene, which that is promoter sequence. This promoter sequence and transcriptional gene, which that is promoter sequence. Promoter sequence will be transcriptional factors, when will be bind with the genes. So this is the genotype, and helicase enzyme used and polymerase enzyme used to transcription to produce messenger RNA, and this messenger RNA is go into the phenotype. So this protein on the basis of desired. So this protein is on the basis of desire will be occur, but the excess of the protein will lead to control by the epigenetics. So the epigenetic is due to the methylation and acetylation on the basis of requirement. So that is the important things. While epigenetic modification we will discuss and how it's very important. So let's begin to understand. First is here is the messenger RNA will be produced. So that is important things. So this is the basically is the direction toward the genotype will express the phenotype. So that is why the genotype will express the phenotype. So the genotype is the whole genome which contain genes for expression and for maintenance of life, while the expression due to the gene is known as the phenotype. So remember about that. So let's begin to understand the epigenetic modification, which that is basically is the methylation and acetylation. So the methylation will be silence of the gene, while the acetylation will be express of the genome. So that's important thing is that histone modification is also occur, which that is allowed to unwind and wind the histone protein with the DNA is known as a histone modification. So the histone modification is the another part which that is known as the histone methylation or acetylation. So here is the DNA, which that is contain a transcriptional gene. So that is chromosome, and here is the inhibition of transcription. So this inhibition of transcription is basically the methylation is here, which that is the transcriptional gene. This transcriptional gene will be transcribed, and that is transcriptional part of protein. This transcriptional part of protein will transcribe to produce messenger RNA, and that will be show. But here is the methylation. Methylation will inhibit of transcription. So it means the methylation will inhibit the from the promoter to the gene sequence which that is required to synthesize the protein will be transcriptional as translational inhibitor. So the translation will be not occur. So that is why the methylation can be harmful or helpful. For example, the cancer causing gene will be inactive due to the promoter region will attach with the methylation, a methyl group. While when acetyl group on the basis of gene mutation, epigenetic mutation, which that is acetylation will be occur, and acetyltransferase enzyme used to transfer, and that acetylation will be activate the promoter region to give the response to produce the messenger RNA, and that messenger RNA will be translate into the protein. So that protein is harmful or helpful, that is depend. For example, the environmental requirement is on the basis of environmental stress or something which that give response, or the cell proliferation due to the increase of the oncogene, and tumor suppressing gene will be inactive. So that means the tumor suppressing gene will be inactive, so the messenger RNA will be produced, and that produce protein. So this protein is mice gene and ras gene will be produced, and that means it will be increase the cyclin and CDK. The cyclin and CDK will be increased. Next we will discuss, and that's lead to proliferation of cell division and increase the cell number, while the DNA damage is more due to the uncheck from the checkpoint of the cell cycle. So here is the oncogenetics, the mechanism of cancer. A tumor suppressor genes and oncogenes, which that is here is the cell division uncontrolled. So that is the normal gene. This normal gene is basically is the tumor suppressor gene. This normal gene is the basically tumor suppressor gene. So this tumor suppressor gene is the oncogenes lead to inhibit the oncogene. So the tumor suppressor gene when inactive, so it will be not control the oncogenes. So the tumor suppressor gene when active, it means the acetylation will be occur. So the oncogenes is basically not transcription access, just normal. So it means when excess of the tumor suppressor gene which that is produce that protein which that is inhibit the production of basically oncogenes, the production of cyclin, the production of CDK, which that is inhibited by the tumor suppressing gene by production of P53 protein and P21 protein. So let's another is thing is that the abnormal gene, if we will discuss, this abnormal gene is the tumor suppressor gene. This tumor suppressor gene will be methylated. When the tumor suppressor gene will be methylated, so the ras and mice genes or oncogenes will be transcriptional factor bind, and this will lead to uncontrolled cell growth. So the proliferation and uncontrolled cell growth without checkpoint to damage the DNA will be not separate. Why? Because the checkpoint is not present and the tumor suppressor gene is used to checkpoint. So that is tumor suppressor gene are not present, so the damaged DNA containing cell is completely cycle and divide, so that apoptosis will be not occur and that cell will lead to the progression of the cancer. So on the other hand, the epigenetics about inheritance we will discuss. If so, the inheritance is the major importance that the cell to cell inheritance. For example, we have multiple cell, the multicellular, which contain a cell contain a undifferentiated cell, basically is contain a methylated after the birth. So this differentiated cell basically are methylated, and that is known as the methylated DNA. This methylated DNA when replicate the whole genome for cell division. So this DNMT1, and that is the ND95 factor will be bind to again more at the methyl group for the daughter cell. So again the maintenance and retention of the methylation will be occur in the somatic cell. So that is known as the somatic maintenance. While in the imprinting is another epigenetic modification, which that is basically this meaning of imprinting same act as parent. So for example, sperm and egg contain a genome. This genome contain a methyl group or a acyl group. So this is the methyl group, and that's methyl group basically is when fertilized the half genome with to become a zygote. This zygote contain a 6 number of methyl group, which as shown in the figure, and that's converted into the embryo. So this is the retention, and this is the retention of the cell. Basically when retention will be removed, so again restoration will be occur and retention will remain constant. While if the retention of the methylation will be decreased or lost, so the next cell division will lead to again methylation. So that is known as the restoration, while the constant number of methyl group remain constant is known as the retention. So remember about that. And last one we will discuss about the epigenetics. Mutation is a transgenerational inheritance. So the transgenerational inheritance mean, for example, the parent is basically contain a methyl group, so that is the erased. After the erased in the daughter cell during their child's, so it means the DNA is without methylated. So the DNMT3A and 3B, which that is lead to again methylation. So that is known as the remethylation. So this remethylation will be produced. The here is the RNA will be lead to produce, which that is functional, and that's lead to produce in the reproductive system. When reproductive system, meiotic cell division will lead to again remethylation and lead to add in the sperm and egg, and that's go into the parent. So I hope you make sense about that video. Please make sure to subscribe, like and share, ask the question for answer, and comment about the artwork. You can also answer question to each other for solving the problem, and I hope this video was good. And next video I will try to explain more about the epigenetics, and we will discuss about also the epigenetic modification with the detail. So that was the overview. Next we will discuss. So thanks for watching. Bye.